Okay, so I will discuss briefly all the questions that we have discussed today. So the first question is about transferrin. That why do we need transferrin? So transferrin is a moiety that transfers iron in the body, and so therefore the correct option was iron. That it transfers irons, not zinc or any other metal. And transferrin is the only enzyme that is present that transfers iron. And other than that, ferritin is there to store iron, and also hemoglobin and myoglobin are there, which basically does the oxygen binding process. And the second question was about enzymes that what do we call about enzymes that contains metal. And so for the correct option was metalloenzymes, that enzymes which contain metals was known as metalloenzymes, and there are also enzymes which do not contain any metal at all. So there is one example that we have showed here, that is nitrogenase. So nitrogenase is an enzyme that basically break down nitrogen gas to form ammonia, and that is present in nature, and it has iron and molybdenum as the central ion. And this is basically the equivalent of the industrial process or the synthetic process that has been developed by humans. That's about the Haber-Bosch process, which is industrially used to for the production of ammonia. And yeah, ammonia is a very important molecule that is used to produce various other derivatives like fertilizers, and that has been used in commercially as well. Other than that, we have talked about ferritin and how ferritin is being used. So ferritin is the enzyme that stores iron plus 3 oxidation state, and then it further exchanges the excess iron whenever it is necessary to other enzymes depending on the situation. So the function is basically not only to uh, uh, store iron, but it actually regulates the iron in somehow. So it stores the iron in plus 3 oxidation state. It takes the iron from plus 2 from transferrin and stores it in plus 3 and then further it can release it whenever it is necessary. So this is one of the example that we have shown here, that it basically exists as a dimeric form, and there are two iron molecules in the ferritin molecule. And then we have discussed about transferrin and the oxidation state of iron in transferrin. So transferrin has an oxidation state. The iron exists in plus 3 oxidation state, and we have discussed about that here in the active site there are like various moieties like histidine, tyrosine, and aspartyl moieties, which basically binds with iron, and there is also a carbonate moiety which further stabilizes the iron in a hexacoordinated way. So therefore the correct option is plus 3 oxidation state of iron, and uh, and the iron is in a hexacoordinated state in transferrin. But if you see the iron carrier or the iron transport moiety that is used in microorganisms, that is not transferrin, that is basically known as siderophores. As siderophores also has iron in a hexacoordinated way, and here also the iron is in plus 3 oxidation state as just like transferrin. But here you have different side chains that binds with iron. Here you have a citric acid, and then you have this hydroxy containing moiety, that is your 3,4-DHBA, and so therefore the correct answer is heterophores. And then we talked about carboxypeptidase, that what is the active metal center in carboxypeptidase, and what is its role. So basically the active center in carboxypeptidase is zinc, so it has zinc in plus 2 oxidation state in the active site, and you have two histidine molecules in the active site and a glutamate molecule, and there is also one water molecule which basically binds in the with the zinc. And so carboxypeptidase exists in two forms, that is carboxypeptidase A and carboxypeptidase B. But carboxypeptidase A cleaves the peptide bonds, which is your various amino acid long chains from the C terminal end of the peptide bond. So the next question was about what are the metal ions responsible for nitrogen fixation, and we have previously also discussed that it is your iron and molybdenum. So the structure of that molecule is basically where you have this iron in plus 2 and plus 3 oxidation state in the structure, and you have the iron sulfur cluster. Overall, it is an iron sulfur cluster, and you also have a molybdenum in a plus 3 oxidation state. So it basically has two metals, that is your iron and molybdenum. And iron exists in both the oxidation state plus and plus 2 and plus 3, but molybdenum exists in plus 3 oxidation state. And then we asked about there is a question about iron 3 plus in its heterophores. So basically, as I have previously shown in the structure, so it actually is a octahedral geometry where you have a high spin d5 configuration as compared to a low spin d5 configuration that we can figure out from the crystal field theory, that as there are oxygen molecules, so those are weak field ligands, so probably it should be a high spin d5 configuration in iron 3 plus, and obviously it is a octahedral geometry. And then we discussed about the role of carboxypeptidase A, that we have previously also discussed, that it is to cleave the C terminal end of the or the carboxy terminal end of the peptide bond and release amino acids from the carboxypeptide end. So it is an enzyme that cleaves amino acids from the peptide bond, and that also from the C terminal. And then we discussed about the storage of iron in human body. So how is iron stored in human body? So that also is done by ferritin only, and in us also the ferritin is the only enzyme that stores iron in uh, in our body, and hemoglobin and myoglobin binds oxygen, and obviously cytochrome is not present in our body. And then the question is about hemoglobin and myoglobin. So if you see, myoglobin is used for what? Obviously, it is used for a dioxygen binding moiety. So here the structure of myoglobin or hemoglobin basically is a tetramer, and in myoglobin is basically about a porphyrin system, and then you have a histidine moiety from one of the phase binding to the iron, and there is a oxygen moiety from the other phase that is binding to the iron. So it is a dioxygen binding metalloprotein. And then we discussed about which of the following statement of hemoglobin is incorrect, and then we showed that that iron is basically coplanar, that iron is actually not coplanar in the deoxygenated form, and it is actually in a tense state, and because the histidine is basically pulling it out of the planarity, and when the oxygen molecule comes, the iron actually comes in the plane of the porphyrin system, and therefore the idea was to understand this coplanarity in this question, that iron is actually not coplanar in the deoxygenated form. And then we discussed about hemoglobin, about the oxidation state of hemoglobin when it is in the oxyhemoglobin state, and also the oxidation state of the oxygen molecule. And then we saw discussed that in the deoxygenated stage, the iron is in plus 2 oxidation state, and when it takes oxygen, it goes to the iron plus 3, and the oxygen goes to the O2 minus or the peroxy state. And there is also a distal histidine that actually helps the oxygen to get into the peroxy state. And then we discussed about methemoglobin, that in our body itself, due to some other enzymes or some other oxidation processes, sometimes hemoglobin is side by side getting oxy uh, de- is forming methemoglobin. It is iron is getting oxidized from plus 2 to plus 3, and we get a plus 3 uh, form iron, that is methemoglobin, and basically this methemoglobin cannot bind to oxygen at all. So therefore it is a side reaction that might happen due to some other oxidation processes in our body. And therefore the oxidation state of iron is in plus 3 oxidation state. And then we discussed about the cooperative binding of oxygen in hemoglobin and why is it so. And then we understood that the cooperativity is because of the side chain in the protein, of the overall protein. So therefore, when it is in deoxidation state, it goes to the oxide state, there is a overall change in the shape of the whole unit, and that is because the binding with the first oxygen, and that actually is helped by this uh, by the by the other unit, and therefore the more and more oxygen got bound, the more better it is able to bind the upcoming oxygen. So therefore the rich become more rich. So that's the basic idea of cooperative binding. And then comes the thing about the pH dependency of the curves of hemoglobin and myoglobin. And then here we have discussed that myoglobin actually binds oxygen at a much more higher affinity than hemoglobin. But in hemoglobin there is a cooperative binding, so that's why the more the oxygen is available, the more better it will bind. And obviously there is a pH dependency in hemoglobin as compared to myoglobin. So the overall curve of hemoglobin is sigmoidal and pH dependent, and now the overall curve for myoglobin is actually hyperbolic and pH independent. So the correct option is option A. And then we discussed about the pH values. How are the pH that in which pH the oxygen binding efficiency in hemoglobin is the best. So that's your pH 7.4. That is our physiological pH in our body. At this pH, the oxygen binding efficiency of hemoglobin is at the maximum. And then obviously it depends on the various pHs in our body. So if you decrease the pH, there is a release in oxygen, and if you increase the pH, there is a more greater uptake of oxygen taking ability. But the physiological pH is pH 7.4. So far we have discussed almost 20